Hello, students. This is Dr. Faizan Mirza. In this video, I'm discussing with you the chapter of cell structure and the structure that we'll be covering basically here in the first part of this video will be the microscopy. So let's start with this. So when we are discussing cell structure, just try to recall what you already know about cells, what structures are you familiar with, recall their functions and just compare them whether which ones are present in plant and which ones are present in animal cells. And with this done, that's just basic preview. What do we know about cells? Cells, we know they are the basic units of life. The first scientist, Robert Hooke, he was the first scientist to observe these under light microscope, and he actually told the world about the cells and the term the cells. This term that he used, this term actually means compartments. All living organisms are composed of cells. There were various other scientists apart from Robert Hooke, who actually contributed to the cell theory, and eventually all that knowledge builds up to the present day. So we know that living organisms, they could be either unicellular or multicellular. They all are made up of cells. Unicellular, for example, bacteria, for example, malarial plasmodium, which is a protoctist, and plants and animals and fungi, they all are living organisms and they all are made up of cells. So when we're discussing living organism, you need to keep in mind that viruses are not living organisms. We do not consider them as living organisms, so virus cannot be regarded as a cell. So we can never use the term of virus and cell together, at least not in the context that viruses are cells, because viruses are not cells, viruses are simply particles because they are non-living outside the host. As far as the cells are concerned, they are present only in living organisms, which could either be unicellular or multicellular. When we classify cells, we say the cells could either be prokaryotes or eukaryotes. Prokaryote cells generally lack nucleus. The basic most feature is that they lack nucleus and they lack membrane-bound organelles as well, which means that all the organelles that have a single membrane or a double membrane, they all are absent in prokaryotes. But when we discuss eukaryotes, they have nucleus, they have membrane-bound organelles, and single membrane-bound organelle, double membrane-bound organelles, as well as those organelles which are not bound by membrane. A lot more on this a bit later. You need to define what cells are. Cells are basic unit of living organisms surrounded by cell surface membrane. So yes, rather than using the term of cell membrane, we prefer to use the term cell surface membrane now. The genetic material is present within the cell, which is DNA, and then there are cytoplasm and organelles present within the cell. Organelle. How do you define an organelle? Organelle is basically a functional and structurally distinct part of the cell. So two things need to be kept in mind: functionally as well as structurally. It's a distinct part of cell. For example, mitochondrion, or for example, Golgi, or for example, ribosomes. They all are functionally as well as structurally distinct part of the cells. What's the nucleus? Nucleus or nuclei, it's just generally basically a relatively large organelle found in eukaryotic cells, but they are absent in prokaryotic cells, something that we already discussed. This nucleus contains DNA and also control activities of the cell. As this nucleus surrounded by two membranes, and that's why we say this is a nuclear envelope. So nucleus is surrounded by a double membrane. Apart from this, what we need to discuss, we need to recall and understand the term protoplasm. Protoplasm is basically every part of cell which is living part. So what part of cell is non-living? Cell wall is a non-living part of the cell. So protoplasm is actually the whole cell, like the animal cell, since it lacks the cell wall, so it's completely protoplasm. Cell walls are made up of different compounds in different organisms. Bacteria, fungi, plant cells have different kind of cell wall. The biological molecule is different for each cell wall. Cell wall can be removed using enzymes, and whatever is left behind after removal of the cell wall from the cell is called as a protoplast. Animal cells itself is a protoplast because it lacks the cell wall altogether. In our cells, we see compartmentalization, which means that we have various compartments in our cells, and there are some unique features in the cell. For example, cytoplasm, nucleus, they are different compartments. We see division of labor, which means the workload in the cells is divided. For example, mitochondria is having a workload of providing the cell with ATP. ATP is the energy currency molecule of the cell, and it stands for adenosine triphosphate, the energy currency molecule. Where's ATP produced in the cell? We know that ATP is produced in mitochondria. You are very familiar with this. But what cell parts other than mitochondria are serving as the site where ATP can be produced? So ATP can be produced in the chloroplast as well as in the cytoplasm, and obviously the mitochondria as well. When we are discussing cells, we know that cells are not visible to the naked eye because they are very small structures. So microscopes are actually required to study cells. There are two types of microscopes that we study at A level: light microscope and electron microscope. And the technique is light microscopy and electron microscopy. The light microscope uses the visible light of electromagnetic spectrum to view the specimen, whereas the electron microscope uses the electron beam to view the specimen. We'll discuss this in detail. Typical diameters of the three type of cells that we discuss here in this chapter: bacteria, animal cells, and plant cells. Bacteria generally are 1 to 5 micrometer. Animal cells are up to 20 micrometer, could be as small as 7 micrometer for RBC, but yes, up to 20 micrometer. And plant cells are much larger, about 40 micrometers. So you can see all the cells which are bacteria, animal, and plant cells, they all are in the range of micrometers in general. You need to recall the term magnification. Basically, it's the times number. It's the number of times the image is larger of an object than the actual size of that object. So magnification can be calculated using the formula of image upon actual. Yeah, be very familiar with this formula and try to know how to rearrange this formula so you can basically carry out the calculations of magnification as well. Resolution. Resolution is the ability to distinguish between two objects which are very close together. The higher the resolution of the image, the greater the detail can be seen. What this actually means, we'll discuss this in bit detail when we are discussing the limit of resolution a bit later on in this video. So coming on to the cell structures generally present in an ML cell, that can be seen using a high quality light microscope. So what do we see? Inside an animal cell, we see the nuclear envelope, we see the chromatin, which are deeply staining and thread-like. We see nucleolus, which is deeply staining. We see the nucleus. So this entire structure is what basically is visible in the light microscope. Together with this, we can see centriole, which is always present near the nucleus. You can see this is a single spherical structure, which is seen under the light microscope. This is a centriole present in the animal cell. Its role is basically in the nuclear division. Then we have cytoplasm, and this ground substance is cytoplasm. Then Golgi body. Golgi body is this tubular structure, and you can see these tiny circular structures at the end of Golgi as well. But together we call this as Golgi body only. There are some small structures that you just cannot identify because they are too small to be identified using the light microscope. We have mitochondria, several mitochondria are there, and you have cell surface membrane as well. Why are these small structures difficult to identify? Because these structures are difficult to identify since the light microscope which you are using is having a low resolution and low magnification. Anything which is smaller than 200 nanometer will not be visible using a light microscope. Why is that so? Because light microscope uses a smallest visible wavelength of 400 nanometer, which is violet color, the shortest visible wavelength for the human eye, and the limit of resolution is half of it. So anything which is smaller than 200 nanometer will not be visible to you. So all these structures which we cannot see are smaller than 200 nanometer. And the ones that you can very well see in the light microscope are actually larger than 200 nanometer. That's why they all are visible to you. When we discuss the generalized plant cell, we see almost the same details there: the nucleus, mitochondria, cytoplasm. We see additional structure like vacuole, which is central in position and is pretty large. The membrane of the vacuole is called tonoplasm. This cell surface membrane pressed against the cell wall, so this is an additional feature here. In plants, we see neighboring plant cells are there, and the cell walls are just lying adjacent to one another. And the middle lamella is a structure which is present as a thin layer that holds the two cells together, which means it's basically present between the two cell walls of adjacent plant cells. Then we have plasmodesma as well. So this gap here is called the plasmodesma. This gap normally is having an an endoplasmic reticulum running from one cell to another. So this provides a connection or a pathway for substances to move from, for example, this is cell 1 and this is cell 2. So substances can move from first cell to the next cell or from the second cell to the first cell through this plasmodesma. So plasmodesma, this connects the cytoplasm of the neighboring cells. Apart from this, there's cell wall, which is of the neighboring cells. There's cell wall and chloroplast. So this is the same detail here. Chloroplast. So yeah, the chloroplasts are here, and you can see the chloroplasts are much larger than the mitochondria. This is a question that normally is asked. Is chloroplast larger than mitochondria or not? Yes, it's pretty large. You can see it's pretty large, and it's visible in the light microscope as well, similar to mitochondria, but about a lot larger. And there's grana, which is just visible. And this grana is the region where you have the photosynthetic pigments located inside the chloroplast. And then again, you have small structures which are difficult to identify. If I ask you why you cannot view these It's because these structures are smaller than 200 nanometer. And since you are using a light microscope, the light microscope uses a visible wavelength of 400 nanometer as the shortest visible wavelength, and limit of resolution is half of that wavelength. So the half of that wavelength is 200 nanometer. So anything smaller than 200 nanometer will not be visible using the 400 nanometer light, which is the shortest visible light in the light microscope. So with this covered, let's move on to the next thing here. You can see this diagram of human cheek cells at 400 magnification. These structures can be seen. This is the cell surface membrane, and this is the cytoplasm. This the nucleolus inside the nucleus, and this is the nuclear envelope, and this is chromatin, or you can say the nucleus, chromatin inside the nucleus. So you can recall how they look like under the light microscope. So yeah, let's move on. This is the detail of the light microscope. We know that the light microscope, which is being used to study the animal cell or plant cells, which we have discussed right now, the magnification resolution of of this light microscope is low. The magnification is low. The resolution is low. Magnification basically means enlargement of the object and the image, how much the image is enlarged as compared to the object. And resolution basically means how much quality is there in the image, how many details can you see in the image, how good is it. So and in the quality, in the terms of quality, we do not use the term of clarity. We avoid the use of term clarity for resolution. This term should be avoided. So coming to the light microscope. So in light microscope, we have an eyepiece at the top, and there's a tunnel, and at the bottom we have you have an objective lens. The light microscope is having either the eyepiece of 6x or 10x. One of them will be provided to you. The objective lens, although there will be two objective lens, probably you'll have to use one or both, one after the other. One is low power and one is the high power. The low power objective is called 10x. The high power objective is called 40x. This x actually means that this will magnify the image up to these times. For example, this 6x eyepiece together with the 10x low power, together it will basically just magnify the image up to 60 times. And similarly, for example, if you are using an eyepiece of 10x magnification and at high power, so this will magnify your image by 400 times. This is something that you should be able to just connect with, that your magnification in a light microscope is basically a multiple of both lenses, the eyepiece lens and objective lens. Just beneath the objective lens, you have your cover slip, and below the cover slip, you have your specimen. And on the specimen is basically placed on the glass slide, and the glass slide is placed on the stage. So once you handle the light microscope, you basically understand what these references, these structures are. And just beneath this stage, you have another lens, the condenser lens, that collect all the light which is being emitted from the bottom of the light microscope. So when you have the light microscope arranged in this way and you turn the light on, the light basically passes through the condenser lens, it gets focused, and it passes through the specimen, it crosses the objective lens, it travels up in the straight path. And these lenses, these which are there in the light microscope, they make sure that the light travels in a straight path and the light moves to the eyepiece lens. And from the eyepiece lens, you are able to basically view your image of whatever specimen you actually had placed. What kind of specimen can you see in the light microscope? You can see a living specimen and a dead specimen. Either both kind of specimens can be seen. There is no compulsion that the specimen must be dead. There is no compulsion that the specimen must be living. You can view either of these specimen using a light microscope. And what kind of stain can you use? You can use a water soluble stain, an aqueous stain, and this stain will add contrast and it will add to the details of the image. For example, chromatin will appear very condensed and not condensed, very thick in color. Because it will take a lot of aqueous stain, and this is how we use the stain to identify the parts of cells within the specimen. This is what I was talking about. There's resolution and magnification. So the limit of resolution is basically half of the wavelength of that particular source of illumination. It must pass through an object for it to appear clear. This is your entire electromagnetic spectrum. This is the shortest wavelength. This is the longest wavelength. The shortest wavelength is 400 nanometer. The longest wavelength is 700 nanometer. You cannot view any wavelength below 400 nanometer. This is the last wavelength that you can see, the shortest wavelength visible to human eye. Now, The limit of resolution is that half of this must pass through the object for it to appear clear, which is 200 nanometers. So any object which is 200 nanometer or larger, which will be visible to you, but any object which is smaller than 200 nanometer will not be visible to you because it is smaller than the half of the wavelength being used. How do we apply this here? You can see that the mitochondrion is as large as 1000 nanometers. So a light of 400 nanometer will half of 400 nanometers is 200 nanometers. So how many times 200 nanometer can pass through this particular object? Several times because this object diameter is 1000 nanometer. So this is mitochondria. We can see this mitochondria, but ribosomes, these are very tiny structures as small as 25 nanometer. So can a light of 400 nanometer whose half wavelength is 200, how many times can it pass through an object which is 225 nanometer in itself? Obviously, it cannot pass. So yes, this structure, which is smaller than 200 nanometer, it will not be visible to you using the light microscope. And this is exactly where the electron microscope basically comes for use. We use monochromatic light beam in our experiments or in questions. They tell you monochromatic light is used. Monochromatic light simply means the light of single wavelength. For example, when you're using white light, it is having combination of all these wavelengths from 400 to 700. All these are there together. But when you use a monochromatic light, it means you are using a light of a particular wavelength, for example, 600, for example, 700. Let's suppose, for example, if I'm using a monochromatic light of 700 nanometer. In that case, my limit of resolution will be half of this 700, which will be 350. So now if I use this particular light, in that case, I won't be able to see any object smaller than 350 nanometer. Similarly, if I'm using a light source of a monochromatic light of 500 nanometer, in that case, my half of this visible wavelength of 500 nanometer will be 250. In this case, my limit of resolution becomes 250. So any object smaller than 250 will not be visible to me, but all the objects larger than 250 nanometer will be visible using a specific wavelength. So normally we get a specific wavelength in the question and the wavelength is provided to us, and then using the wavelength we have to just compare that whatever we are seeing, the diameter of that is it is smaller than that or larger than that. So if the diameter is larger than that, then definitely it will be visible to us. If it's not, then it will not be visible to us using that particular wavelength. How the wavelength affects the magnification wavelength. Is there any connection of wavelength and magnification? No, there is basically not really a connection between wavelength and magnification because as you increase the wavelength, the magnification is not affected. Magnification is primarily because of the lens. But how is the resolution affected? Resolution is basically inverse of your wavelength. The wavelength increases and the resolution decreases. So if you use a shorter wavelength, so in that case, your resolution will increase. Moving on from you just recall these magnification formulas. Magnification is equal to image upon actual. You can rearrange the formulas for actual and for the value of I, and you can just use these in your calculation. So yes, keep this formula in mind as well, that your wavelength is inversely proportional to resolution. This actually helps you in a lot of MCQs where you are expected to answer something. They are providing you some few wavelengths and values are given to you, and you are asked to just say whether resolution is basically changing or not. And wavelength, and as far as the wavelength is concerned and magnification is concerned, wavelength and magnification have no real correlation. So if you change the wavelength, the magnification of that particular image will stay unchanged because magnification is basically dependent on the lens and how many type of lenses are being used. Are you using a 6x objective or a, oh, sorry, 6x eyepiece, or are you using a 10x eyepiece, or you are using a 10x objective or a 40x objective, for example. So moving on from this, let's move on to the last part of this video where I'm discussing electron microscope with you. Electron microscope basically is like a long, tall tunnel in which you have an electron gun at the top. This electron gun is basically heated up to produce a beam of electron that travels in a path. And inside the electron microscope, you have vacuum to make sure that electron beam does not get deflected. We have electromagnetic lenses, and all these electromagnetic lenses are there to ensure that the electron beam keeps traveling in the straight path. We place our specimen somewhere here. The specimen must be dehydrated, which means we cannot use a living specimen. Since the specimen is dehydrated, so all the water has been driven off. In that case, the specimen will not be a living specimen. Now, if you pass the electron beam through it, the image will be basically seen on the screen at the bottom. Now, why we use a dehydrated sample? The dehydrated sample is used because in a vacuum condition, water evaporates at room temperature. So if you kept a living specimen here and there was vacuum inside, and in that case, all the water in this particular specimen will basically fog up in this entire electron microscope. A lot of mist will be created, and it will start to affect and in fact it will start to deflect the electron beam in some random direction, which obviously you cannot allow. Electron beam must pass through the specimen straight. That's why you are using electromagnetic lenses here, so that the electron beam travels in a straight path. You use heavy metals, and these heavy metals allow you to uh, add stain to the specimen. The image that we get here is basically a grayscale image. We do not see a colored image. The electron beam that we are using, it's having a wavelength of zero point 0.1 nanometer, and the limit of resolution becomes 0.05, just half of the wavelength which we are using. The magnification is very high, up to 100,000 to 250,000, achievable. Now, when we discuss electron microscope, there are two types of electron microscopy that we discuss in our syllabus. One is called TEM, which is transmission electron microscopy. This for internal details, something that we have just discussed. Thin sections of specimen are used here. The other one is scanning electron microscopy, or SEM. This basically is used for surface feature study. Very thin sections are not required, and it ends up producing 3D images of the specimen that we are using. So with this, I'll wrap up this video. I hope this was helpful for you. That's it from my side. Thank you so much.